computing. Turing machine is kind of like what you think a computer is. You give it a problem, you write it down on a tape, and then you go away. You don't interrupt it while it is working. It sits and thinks for a while and thinks in a very mechanical way. It eventually halts and gives you an answer. Or it doesn't halt. It goes into this loop and just keeps stuck in there forever, right? But you can't interrupt it. Well, brains are not like that, right? I'm constantly being interrupted. I'm talking to you, your brain is receiving input. So, at the very sort of basic conceptual level, a Turing machine is not fitting as a model of the brain. So the next time people tell you the brain's a computer, do a double take, think about Turing machines. Now, here's the irony. Most of the things that we call computers also take inputs. If you're typing away at them, they're attached to the internet. So the Turing machine is not a very good model of operating systems or all these things that computers do. So, in fact, computer scientists have been exploring so-called models of interactive computing, non-Turing models of computing. Now, here's the interesting thing. Turing himself thought about the brain a lot. That's how he came up with the notion of the Turing machine in the first place. He took a year off. He was very successful. He cracked the code, cryptographic code, during the war. And he got a bit of time to himself. He took some time off. And then he wrote this paper called Intelligent Machinery in 1948. This was amazing. He came up with the idea of neural networks, of genetic algorithms, trainable neural networks, and so on and so forth. He took this paper to his supervisor, and the supervisor heavily criticized him. There were some smudges on this paper. It was not well typed. Didn't like that. And these are some comments that the supervisor made. He said, a bit thin for a year off. Schoolboy's essay, not suitable for publication. Poor Turing never published this paper. So when somebody discourages you about your research, keep in mind that other people have been discouraged as well. If you have conviction about it, go for it. But don't fool yourself, of course. So Turing never published this paper, which passed away. It was published posthumously. It is available freely on the web. Please go read it. It's fun to read. So brains are not computers in the sense of Turing machines. And this is, in fact, what makes brains so interesting to study. We actually don't have good models of what brains are, how to think about them. But brains are, if you want, intelligent machines. So one reason to study brains is to find out how nature designs intelligent machines. How does the process of evolution come up with intelligent machines? Now, people have been studying the brain for a while. It's not a new enterprise. And the last 100 years have seen some pretty profound advances, but we are still at early stages. It's kind of like, in some ways we know a lot, but in some ways it's like chemistry in the days of alchemy. We don't quite know how to think about it. There are many theories. It's not like there are not theories. One theory which has been very useful is the so-called place theory. And the place theory basically says that different parts of your brain do different things. That's a very simple theory. You've got a visual part of your brain that is useful for seeing. Theory goes back a long time ago. People had ideas about phrenology. They would read bumps on the skull. And this is an illustration from the 15th century. You can see, I guess this illustrator thought that memory lived back here. But, okay, imagination lived here. Now, we've moved away from that. But the place theory still holds sway. How many of you have heard of Phineas Gage? Some of you. So there was the strange case of Phineas Gage. There was an accident. He was working, I think, to lay down a railroad. And this iron rod that he's holding, there was an explosion, and this rod went straight through his head, landed on the other side. You can see here a reconstruction of how the rod went through his head. He was still actually conscious, walked to the hospital or something like that, and then collapsed. So it was amazing because there's some other parts of the brain where if you knock it out, you're not going to be conscious and walking to the brain. Walking to the hospital. One of the remarkable things that people found out after this accident was Phineas Gage, even though he otherwise seemed fine, he started having these personality differences. He was a different person from who he was before he had had that accident. And so even something as, if you want, seemingly fuzzy or ill-defined as personality traits, could be localized in some parts of the brain. That's what people realize. And we've been learning about the brain one lesion at a time. If you have a stroke or some damage, you might lose a particular function, right? So there is something to this place theory. However, it's not a very insightful theory. It's a useful theory. Because the brain is really a circuit. So thinking about the brain in terms of places is a bit like thinking about a country in terms of the cities, but not thinking about the roads that connect these cities. And even though I kind of ruled out the computer as a good model, at least the Turing model as a good model for the brain, nevertheless, it's something like that in the sense that it's made up of circuits and parts. And what it does, whatever its function is, comes out of this circuitry. And we don't know what those circuits are for the most part. After all these years of research, our knowledge of brain circuitry remains quite incomplete in a way that I will illustrate for you in a minute. But let me first say that there's some part of your nervous system circuitry that we do understand well, and that's your peripheral nervous system. This is the part of the nervous system that talks to the rest of your body, to the viscera, and a lot of medicine is of course predicated on understanding this peripheral nervous system. But the central nervous system, namely most of the stuff that's inside the cranial cavity, for the human brain in particular, our knowledge is really very limited. Francis Crick, one of the co-discoverers of the structure of DNA, towards the late parts of his life, got interested in the brain, and he wrote this paper with Keck Jones. Both Crick and Jones have passed away, complaining about the backwardness of human neuroanatomy. And the odd thing is, it's not just humans that we don't know brain circuits for. Even the best studied laboratory specimen, species that we study in neuroscience, which is the rat, we actually know quite little. Here's sort of a representation of the connection matrix, if you want, of the rat brain at a fairly coarse level of resolution, something that I call the mesoscale resolution. I don't want you to look at the details. All I want to look at all the gray. The gray is where we don't have information. So I'm not going to go into detail, but it just tells you there's a lot of places where we don't have information about connectivity. And now contrast that with the genome. So, of course, now we have lots of knowledge about the genome. The human genome has been mapped. The rat genome has been mapped. Just today I read that the coelacanth genome has been mapped. So lots of genomes have been mapped, but not many brains have been mapped in that way. There is exactly one species for which we have more or less a full circuit diagram at some level, only for one or two individuals, and that's the worm, the C. elegans. It's got a few hundred neurons. So early days in that sense. And that's a gap that struck me. I'm a theoretical physicist by background, so I like to think in terms of big pictures, and that's sort of a gap that really struck me, and I set out to try to address it. Now, just to give you a sense of scale, that's the human brain. The mouse brain right there. It's about 1,000 mouse brains will fit into one human brain. The human brain's about a liter, and mouse brain's about 1 cc. Mice are pretty intelligent. That's macaque, monkey, hopper in between. So let's look inside a cross-section of a mouse brain. So this is, I'm going to zoom in into this cross-section. And as I zoom in, you're going to start seeing neurons. So this is the part of the brain that's called the cortex. And what you are seeing here are individual, these dark bodies. They are the cell bodies of the neurons. That's where the nucleus sits, a lot of the cellular machinery. Now notice all this white space between the neurons. Can anyone tell me what's going on in that white space? What's there in that white space? Myelin, so it's myelinated. And what is myelinated? There's glial cells. So what is myelinated? Axons, right? So most of that white stuff is where the wires are. You can't see that in this picture, but let's go to another cross section. This time the myelinated fibers are actually visualized. So not all axons are myelinated, but some axons, wires, have got this myelin sheath on them, which we can visualize. And again, we are zooming in. You will see, this is now we are back in cortex. You can see myelinated axons running in a crisscross pattern. So there are cell bodies, and then there are wires connecting these cells. And in fact, to get a little better feel for that kind of white space, let's look at an electron micrograph. So here again, you see the cell bodies. Now you see the gray stuff. We are now zooming in more and more. And you'll start seeing the little fragments of the axons and the different bits and pieces of the cell. So this is what that white stuff is made up of. It's like a dense spaghetti of wires, a very dense spaghetti. And it's very difficult actually to imagine the complexity of the structure. So I have made a little mental picture for you. So let's think about the human brain. And let's try to zoom it up until you can start seeing the individual fibers. Now the myelinated fibers are a little thicker. Let's go to the unmyelinated fibers, which are a little thinner, between a tenth of a micron to a micron. And let's zoom out the human brain until we can see those. It turns out you have to zoom out the human brain to about the size of a football field before you can do that. Okay, because these are very fine wires. So if you did that, now imagine something about a football field size, both in lateral dimension and in height, and now you're barely able to see the fibers, right? So it is really that complicated. If you were to just take a walk through it, going from one part to another, you will lose track about what's going on. If you did that with the mouse brain is a lot smaller, so you won't need the football field, but you need something the size of this room. So think of something that big, and now you can just see the wires, right? So that's in some sense why it's so complicated to understand. It's got all these parts to it. And unlike the other parts of your body, like your liver or your muscles, there are these interesting cells called neurons which send out very long wires. So what the brain does is really coming from the connectivity patterns of the brain and coming from these long wires. Now I want to again help you visualize how long these wires really are. This is a neuron in the mouse posterior parietal cortex that has been traced. Now, that scale bar here is a millimeter, so this is most of the mouse brain actually. So the neuron has its cell body there, and it's sending these branches far away to long distant parts of the brain. If we go to your motor cortex, somewhere around here, there are neurons that are sending axons down to your spinal cord. And in fact, like just one synapse away, you get to your hands. So these very, very thin, very, very, very long wires, and a dense tangle of them is what the brain is made out of. There's also local circuitry. Now, we can't really visualize these wires in humans. We can do them in non-human animals because the technique that is needed to visualize these wires is invasive. So nobody has actually seen a full-sized human neuron. I would say that seeing one of those would be like going to the moon. It strikes you as crazy. Well, you see the human, you see the human brain, you can see that postmortem, but to visualize a neuron, to fill it up, you can't really do that yet. So I had proposed some years ago with a group of scientists that we need to systematically